Hello and welcome everyone. How are you all? I welcome you to the class of biology. Dear students, please report your attendance and we will begin right away. Okay. Hello. Hello Shillong students, how are you? Hi. So dear students, please report your attendance. That's why I read the long hi. Hello everyone, how are you? Okay, nice to see everyone. Yeah. Centers, please report your attendance. Okay. All right. Thank you. So the other centers, they will also report attendance, and meanwhile I can start from where I left in the last class. So dear students, we have been dealing with the living world, the biological classification, and in the last class I had discussed with you the importance of biological classification, because biological classification is the way in which we can better learn about the organisms. We can categorize these organisms into categories, and categories are much easier to understand. Categories are much easier to understand. Just like we give our friends some nicknames. So in a similar manner, remembering the categories helps you to identify more and more organisms and place those organisms into that category. All right. So biological classification started with Aristotle, followed by Linnaeus, followed by Haeckel, then Copeland, and then finally the Whittaker system of classification, the 5-kingdom system of classification. It has been slightly modified in the form of 6-kingdom classification given by a great scientist called Carl Woese. So he gave the 6-kingdom system of classification also, in which Archaebacteria became the 6th kingdom. Other than that, there are the 5 kingdoms are normal, like kingdom Monera, Protista, fungi, bacteria. Sorry, kingdom Monera, Protista, fungi, Plantae, and Animalia. So we are going to take these kingdoms now. Okay. So in the last class, we had done a few parts regarding this. So I hope you remember. All right. So the biological classification that we were dealing with, biological classification, I told you that it is started with first scientific system of classification given by I had already told you this, classification was by Aristotle. Okay. And thereafter, we have some classification system like 2 kingdoms classification system. It was given by Linnaeus. 3 kingdom classification system was given by Haeckel. 4 kingdoms classification system was given by Copeland. And finally, the 5 kingdom system of classification, it was given by Robert Whittaker, R.H. Whittaker. R.H. Whittaker. So Whittaker had some criteria for the classification of organisms, and this we have already discussed, right? So you remember that? Okay. So Whittaker had some criteria of classification, and we have already discussed it. And Whittaker system had some criteria. So regarding these criteria, I have given you the details. I hope you have done your homework. Whittaker's criteria, it is very important for your board examinations. Okay, we have covered this in the last class. And after that, we had discussed about the 5 kingdoms. These are Monera, Protista, fungi, plantae, and Animalia. So kingdom Monera was what we were doing in the last class, and thereafter we will cover kingdom Protista, fungi, plantae, Animalia. So in the last class regarding kingdom Monera, I had given you few details that these are primitive organisms. Archaebacteria are included in that kingdom Monera, and we have other organisms also. So before we begin that, I'm going to give you a short classification taxonomy slide show. So please watch this first, and then we will start right away. Okay. So classification and taxonomy, I am just doing it again. What is the purpose of classification? It is a grouping of information based on similarities, and there are about 1.7 million species described in both plants and animals, and there are many million species that still need describing, so we do this taxonomy for that purpose. Okay, and it is based, the modern taxonomy is based on the evolutionary relationships. That is, more primitive organisms have given rise to more advanced organisms. This is what we are going to study. Early taxonomic system was given by Aristotle, followed by Linnaeus' system. Okay, so Linnaeus' system was binomial nomenclature and had some other rules also. So these rules we have already discussed. You remember that? Okay, and he gave the Latin language as the medium in which scientific names are to be written. Okay, so yeah, you can maybe most people will recognize the picture of the human being I have labeled there. He's Ronaldo, none other than. Okay, so there are some of the things where we include some third category called the subspecies or the variety. So it is also used sometimes, and this is just a detailed slide. I will give you this slide to you, slideshow to you. Okay, and modern taxonomy is based on a lot of things like chromosomal structure, reproductive potential, biochemical similarities, embryology, breeding behavior, structural similarities. Okay, and 7 taxonomic categories are in this order. Okay, so you have seen all this. All right. So 5 kingdom system, that is what we follow today. So these are 5 kingdoms. Okay, I have drawn yeah, 5 castles, 5 kingdoms, 5 kingdoms of Monera, Protista, fungi, plantae, Animalia. Okay, and it is also categorized into 3 domain system or 6 kingdom system. That also we have done. Okay. So this part we have done in the last class, and phylogeny is a very important term for your board examination. What is phylogeny? It is the evolutionary tree representing the evolutionary history of a species. Okay, so these things and other things as well, keys as well, I have given you. So today we are going to cover the first kingdom. We have to going to finish it today, kingdom Monera. So these are some monerans in this figure here. This is 4,700 times magnified. This is these are bacteria. These are bacteria found in milk, lactobacillus. So it is magnified about 4,700 times. So that is how it looks in this figure. Okay, and the full details of kingdom Monera, I have a separate slideshow. I will give you that also. Okay, and just take a quick revision of this. Kingdom Monera, Archaebacteria and Eubacteria. Their cell walls are not, Archaebacteria are not made of cell wall not made of peptidoglycan, methanogens, halophiles and thermoacidophiles, and Eubacteria are the most common, E. coli, Streptococcus, Cyanobacteria, etc. So the first 5 kingdom of eukaryotic, the first eukaryotic kingdom is Protista. So amoeba, paramecium, slime molds, euglena, diatoms, dinoflagellates, all this will be included in Protista. Okay. Then fungi, So we do not see these beautiful mushrooms in our country. These are European mushrooms. So it's a different species, but it's yeah, it looks like a good burger, right? Yeah, it looks like somewhat like a burger, and somebody wants to eat it, but actually it's not exactly edible. Okay, it looks edible, but it's not edible at all. Okay, so these are fungi, and then plants and animals. So we will cover this today. So just wanted to show you this. This will be helpful in your taxonomy. All right, so let's continue our kingdom Monera. Kingdom Monera. So regarding kingdom Monera, in the last class we have covered that there are many ways to classify bacteria. There are many different ways to classify the bacteria. So regarding that, if bacteria has flagella, we call it monotrichous. If it does not have flagella, we call it atrichous. If single flagella is there, monotrichous. If multiple flagella and all around flagella, it is called peritrichous. Both flagella, one on each side, amphitrichous. Then a bunch of flagella on one side, a lophotrichous. Okay, so all these things we have discovered in the last class. Then regarding the shape of bacteria, if it is round, we call it coccus. If it is long, we call it bacillus. All right. So coccus, bacillus, vibrio, spirillum, all these shapes we have discussed in the last class. And streptococcus, chain-like, staphylococcus, bunch of grapes-like structure. Okay. So all these things we have discovered in the last class. So let's continue. In the last class, I had also discovered a special. I had also told you about a discovered special technique of classifying and identifying bacteria, and this was discovered by Hans Christian Gram. H.C. Gram was a scientist who discovered it. It's called Gram staining. So in the last class, I had shown you Gram staining, how it is done. In Gram staining, I take a glass slide and I fix bacteria onto that glass slide. I pour Gram's stain or crystal violet stain on it. It's a purple colored dye. And when I stain this glass slide with the bacteria on it, the whole slide bacteria become purple in color. But to distinguish one type of bacteria from the other, I pour some alcohol over it, and some bacteria become colorless. Okay? These bacteria which have become colorless are now colored by a second stain called counter stain or saffron. So these colorless bacteria will become pink colored. Already we have purple colored bacteria. Purple color will not get a lighter color. It will not be pink colored. So purple will remain purple. Purple and pink. The purple colored bacteria are called gram positive bacteria. The pink colored bacteria are called gram negative bacteria. So this is a very important system of classification of bacteria, that eubacteria are classified into two categories, positive and negative, gram positive and gram negative, right? So the thing is, yeah, just a second. Okay, so gram positive and gram negative bacteria, there are two types. So I had asked you to do some homework and give me the differences between gram positive and gram negative bacteria. Have you done it? Differences between gram positive and gram negative bacteria. Have you done your homework? Yes or no? Dear students, please share your responses. Gram positive and gram negative bacteria, what are the difference? What are the differences? Nobody? Okay, I am waiting for your responses. All right. Nobody is answering. All right. Look, there are a lot of differences between gram-positive and gram-negative bacteria. I'm just asking you the differences. What do you know about the differences between gram-positive and gram-negative bacteria? The bacteria, yeah, it's okay. Yeah, the bacteria that absorbs the stain is gram-positive. But what is the basic difference between why does it absorb and the other one does not absorb? Okay, you have not done your homework, so I'll have to do it for you. Look, the gram-positive bacteria becomes stained because it does not have lipids. Okay, and it does not lose color because it does not have lipids. Both bacteria absorb the stain, both bacteria. But the difference is when we pour alcohol, the lipids get washed away. Lipids are present only in gram-negative, not in gram-positive. So what this means is that in case of the gram-positive, gram-negative bacteria, because one has lipids, other does not. So one will lose color, other will not lose color. Okay, so you can write this. They retain the stain during gram staining. First point, this will be they retain the stain of crystal violet. They retain the crystal violet stain during gram staining. During gram staining. Gram-negative bacteria do not retain the stain. So this will be common. This is a very easy point. They do not retain the gram stain or the color. They do not retain the crystal violet stain during gram staining. Okay. Second thing is they have a thick membrane. They have a thick membrane. Their membrane thickness is more. They have a thick membrane. Membrane thickness is very much high. And here they have a comparatively thinner membrane. Thinner membrane. Okay. So these are some differences. And so please keep on writing. I'm moving on to the next page. Okay. So yeah. All right. If you want to know the thickness, they have a thick membrane about 20 to 30 nanometer. Sorry, 20 to 80 nanometer. It is very variable. But in case of gram-negative bacteria, it is only 5 to 10 millimeter. Sorry, nanometer. I forgot. It's nanometer. It's nanometer, not millimeter. Millimeter is quite large. Okay. So if it was so thick, we could have easily observed it. But it is not the case. Okay. They have a smooth cell wall. Smooth cell wall. Their cell wall is wavy. Cell wall is wavy. Okay. Fourth thing is um, lipopolysaccharides or lipids are absent in cell wall. Lipids are absent in cell wall, or they are, or very less. If they are present, it is very less. Okay, you can basically say it is having no lipopolysaccharides at all. But in case of the gram-negative bacteria, the lipid content is very high. Cell wall has high lipid content. Has high lipid content. It has a high lipid content, right? Other than that, their cell wall has contents of teichoic acid. Okay. All right. They also have an outer membrane present. Outer membrane present. Here, outer membrane is absent. Wait a second. Outer membrane is normally not present. Wait a second. Do not write this. It's not always the case, so it's better we write other things. You can say teichoic acid is present in cell wall. Teichoic acid present in cell wall. Present in cell wall. Teichoic acid is absent. Teichoic acid absent in cell wall. It's a type of carboxylic acid. So this is about it. There is outer membrane in gram-negative bacteria, and in gram-positive bacteria, outer membrane is absent. There are some other things also. Porins are present. So if you want to write, I can write about 10 differences, but I'm not sure you will be able to write and remember all of them. So I will just conclude this with some examples. Example is Bacillus species is normally gram-positive, Bacillus subtilis. Okay, and Streptococcus species. Okay, Lactobacillus. These are gram-positive bacteria. Okay, Vibrio cholerae, cholera-causing bacteria. Acetobacter rhizobium. Rhizobium is very important because it has been asked in your NEET examination that rhizobium, the nitrogen-fixing bacteria, it is gram-positive or it is gram-negative. So actually it is gram-negative. Okay. E. coli. E. coli is a very common bacteria. It is gram-negative. Okay. So Acetobacter aceti, which makes acetic acid, it is also gram-negative bacteria. So there are a lot of examples. So This will be sufficient for you. You must at least know these 5 differences. I can give you more differences, but I think this will be sufficient. So at least try to remember these 5 points. These are very important. All right. Now regarding the eubacteria, we have to do some other examples also. I hope everybody is finished, right? Okay. Moving on. There is a type of eubacteria called the cyanobacteria. Cyanobacteria. It is a subcategory of eubacteria or blue-green algae. Blue-green algae or BGA. Okay. So these are blue-green algae or blue-green cyanobacteria. So what do these do? These bacteria are photosynthetic and oxygenic. Photosynthetic and oxygenic, oxygen producing, oxygenic, containing chlorophyll. Containing chlorophyll. Okay, so the type of chlorophyll they have is chlorophyll A is present in them, and chlorophyll A is the type of chlorophyll which is responsible for evolving oxygen. Chlorophyll A. Chlorophyll A is found in higher plants normally. Okay, it's found in higher plants normally. It is found in higher plants, and these bacteria have a blue-green color. These bacteria or the BGA, one and the same thing, have blue-green color. Blue-green color due to blue-green pigments. Okay, and these pigments are phycocyanin and phycoerythrin due to blue-green pigments. Blue-green pigments found in cyanophycean granules. Cyanophycean granules in cytoplasm. They have some granules found in their cytoplasm. And these are called cyanophycean granules. So they have a particular blue-green color because of these cyanophycean granules. Like this. Okay. Also, these bacteria are very important. Very, they are highly economically important. The blue-green algae, the BGA, have very high economic importance. Have very high economic importance. have very high economic importance, since they have the ability to fix nitrogen in soil. Because they have the ability to fix nitrogen, because they have the ability to fix atmospheric nitrogen. Atmospheric nitrogen. Right? So they can convert nitrogen into nitrates. Nitrogen can be converted into nitrates by blue-green algae. Okay. And this process occurs in the form of special cells found in the blue-green algae, and these blue-green algae cells are called heterocysts. Normally, the blue-green algae are lying like this. But they have in the middle some large cells with a very thick cell wall, called heterocyst. Called heterocyst. So I'm just coloring it like this. So just for this purpose, for the blue-green color, denoting that color. This cell is called heterocyst. The heterocyst has very thick cell wall. Very thick cell wall. And these thick-walled cells are specially suited to fix nitrogen. Okay. This is a filament of nostoc. Nostoc filament. Okay. So in CBSE, the question is asked, which cells in nitrogen fixing bacteria or blue-green algae can fix nitrogen? Actually, every cell can fix nitrogen, but there is a problem. The enzyme which fixes the nitrogen into nitrates, it can function only in the conditions of oxygen deficiency. So normally the blue-green algae are kept in air like this. So if they fix nitrogen, try to fix nitrogen, the air oxygen will not let the enzyme function. So what they do is they develop special cells with very thick cell wall so that oxygen cannot enter these cells. These cells will be capable of fixing the nitrogen at a very high rate, whether it is in air or below the soil surface. Below the soil surface, where there is no oxygen, every cell is capable of fixing nitrogen. But near the soil surface, where there is sunlight, where there is because these cells will remain near the sunlight. Normally, where there is sunlight, there will be oxygen also, and these are oxygen evolving bacteria. So normally, only the heterocyst will be able to fix nitrogen. Otherwise, every cell is capable of fixing nitrogen. So please write. Next point. Heterocysts are spatial cells with very thick cell walls and are capable of fixing nitrogen. Okay. So heterocyst is a spatial cell capable of fixing nitrogen. This is an important question for your NEET exam, so please mark it separately. Okay? It's a spatial cell capable of fixing nitrogen. All right, so this is about cyanobacteria. And there is one more important thing about cyanobacteria. Please write it under a separate heading, economic importance of cyanobacteria. Subheading, economic importance of cyanobacteria. So the cyanobacteria are capable of not only fixing nitrogen, they are also capable of acidifying the soil. First point is they are important nitrogen fixers in soil, and also release oxygen so they are photosynthetic nitrogen fixers. Second point is they can acidify the soil in which they are growing. They can acidify the soil in which they are growing. So what does this mean? They can turn the alkaline soil into fertile soil. This is a very important point. Therefore, they are used to restore fertility of soil. Now you'll ask why? How? They are used to restore fertility of soil, basically alkaline soil. Fertility of alkaline soil. Look, there are some basic principles. In soil, if we collect the soil of very fertile environments like forest soil from all the fertile regions around the globe, normally forest soil is the most fertile soil. So looking at the samples, the scientists have discovered that most fertile soil of like Amazon rainforest or most other rainforests of the world, which is very fertile, it has a pH of 5.5, acidic pH. And all fertile soils have been discovered to have acidic pH only. So this means that soil pH has close relationship with the fertility power. Therefore, in order to restore fertility of soils which are not able to grow anything, if we measure the soil pH level of infertile soils, normally they are alkaline soils. They have pH more than 7. So we need to make it acidic first. Then we can grow anything inside it. So acidifying property is found in blue-green algae. What we do is take a field and fill it with water and allow the blue-green algae to grow there for 2-3 years. In 2-3 years, they will be able to sufficiently acidify the soil area. That is how that is done. Alkaline soil is infertile, and it's also called ooser soil. Ooser soil or infertile soil. So they help in restoring the fertility by decreasing the pH of the soil conditions, soil environment. All right, so I hope you have done this. Examples of cyanobacteria: nostoc, Anabaena. Anabaena is quite common, okay, etc., etc. There are so many, but these two you will remember: nostoc and Anabaena. Anabaena cycade is found in cycas. Anabaena azole is a symbiotic blue-green algae found with Azolla fern. Example: Azolla. I'll write this later on also, don't worry. When I will teach you Azolla, I will mention the name of Anabaena once again. Now there are some other types of kingdom Monera members, and these are called mycoplasma. Okay, so you have completed this, students? Okay, so next point, please write mycoplasma. Mycoplasma. Okay, so what is mycoplasma? Mycoplasma is a group of organisms which are not having any cell wall. They have triple layered cell membrane. They grow on steroid-rich medium. They cannot survive in a medium without steroids, without cholesterol, and they are normally quite attached to their particular host species. So normally they are quite attached to the host species. They cause a lot of diseases, and they are the smallest living organisms, smallest living cells. They were first, it is also called PPLO, pleuropneumonia-like organisms. They were first discovered by Nocard and Raux. They were first discovered by Nocard and Raux in pleural fluid of cattle, suffering from pleuropneumonia. So that is why it is called pleuropneumonia-like organism. Pleuropneumonia-like organism. Like organism. All right. Next thing is they are the smallest living organisms. They are the smallest living cells. They are the smallest living cells. The world record, the Guinness Book of World Records, is in the name of Mycoplasma genitalium. Mycoplasma genitalium. If question is asked that mycoplasma smallest living cells, okay, but which is the smallest mycoplasma? Then the answer is Mycoplasma genitalium. It is very, very small. It can pass through bacteria-proof filters, causes is the smallest living cells. First of all, let's write it's the smallest living cell is the smallest living cell it is the smallest living cell and causes sterility in human males. It is responsible for causing sterility in males with immunocompromised condition. Weak immune system people can suffer from this. So this has been discovered to be the smallest living cell in all the living organisms. Okay, this is the smallest living cell. If a question is asked general, you can say mycoplasma. If among mycoplasma, then this is the smallest living cell among all the organisms on earth. Okay, moving on. Okay, students, do you need a break? I believe. Do you need a break right now? Raise your hands if you want. Okay. All right. Okay, take a quick break and then we'll return after your break. Okay, when I see everybody has seated up, I'll start the class. Okay, take a quick break, 5-10 minutes. Okay, so everybody is up now. Finished your snacks, I believe. Okay, so let's get started again. So I was telling you that mycoplasma is the smallest living cell, and among all the living cells, mycoplasma genitalium is the smallest mycoplasma. The question that is asked in your examinations is that what are the specific diseases caused by mycoplasma? One question I've already told you, this one. The other question is regarding their habits and their structure. So please write some few points about mycoplasma. Mycoplasma do not possess cell wall. Mycoplasma do not have cell wall. They do not have a cell wall. This means that any antibiotic which inhibits cell wall formation, it will be ineffective against mycoplasma. Antibiotics kill bacteria, and most bacteria, they have a cell wall, right? Nearly all bacteria have cell wall. So if we inhibit the cell wall formation, the bacterial growth will be limited or the bacteria will die. But mycoplasma is specially adapted. It's not having a cell wall at all. It does not have a cell wall. So it means it can survive without it. So any antibiotic like penicillin, which causes cell wall inhibition, in that case, the penicillin will be totally ineffective against mycoplasma. We'll have to give it a separate antibiotic of different kind against mycoplasma. Do not have a cell wall. Mycoplasma do not have cell wall. Therefore, cell wall inhibiting antibiotics are ineffective. Cell wall inhibiting antibiotics are ineffective against mycoplasma. Example, penicillin is ineffective against PPLO. Against mycoplasma, because penicillin is a cell wall inhibiting antibiotic. It was the first antibiotic, and it has the ability to inhibit cell wall growth. Sorry, cell wall synthesis. All right. Okay. One more thing. Mycoplasma require steroids for growth. So everybody knows steroids, right? So it seems like mycoplasma has joined a gym, okay, a microbial gym. So he goes to the gym and he requires steroids for growth. So if steroids are not present, it will not be able to grow at all. Steroids for growth. So they cannot be grown on synthetic medium without steroids. They cannot be grown on synthetic medium without cholesterol. Cholesterol is a type of steroid substance. Cholesterol has a steroid nucleus. So cholesterol is required. If cholesterol is not present or steroids are not present, mycoplasma cannot be grown on synthetic medium. Synthetic medium means artificial growth medium which contains all the nutrients for growth. Okay. Normally it contains glucose, fructose, amino acids, vitamins, minerals, things like that. Okay. So all other organisms can grow, but mycoplasma cannot grow. Mycoplasma give fried egg appearance to colonies. Mycoplasma give fried egg appearance to colonies. Fried egg appearance to colonies. To their colonies. Their colonies look like fried egg appearance. And mycoplasma have triple layered cell wall. Have triple layered cell wall. Sorry, cell membrane, cell wall. Sorry, cell membrane, not cell wall. Cell wall is absent. Cell membrane or plasma membrane. It's triple layered. So their outer coating is triple layered. Now for the final thing. They cause some specific diseases. They cause diseases in both plants and animals. But their plant diseases are more common. So I am writing some diseases which are important from exam point of view. Bunchy top of papaya caused by mycoplasma. Bunchy top of papaya. First disease. And witch's broom disease of plants. Normally brinjal suffers from it. Witch's broom disease. Witch's broom disease happens when the plant leaves that are infected, they give appearance of the sticks of a broom. Okay, so just like a Harry Potter's broom, the plant becomes broom shaped because the leaves are undergoing withering and all that. So it happens normally in brinjal and chilies of plants. A lot of plants suffer from it. Then there is a leaf curl disease of plants. Leaf curl disease. But bunchy top of papaya is very much asked, and witch's broom disease are most asked. Leaf curl disease and sterility in human males. This is animal disease. And PPLO causes pleuropneumonia in cattle. So these are animal diseases. Sterility in human males and pleuropneumonia of cattle. These are the diseases caused by mycoplasma. Pleuropneumonia of cattle. These are the diseases caused by mycoplasma. So in NCERT it is given very less. I have expanded and included all the questions that have been asked in previous year examinations. So this will help you out regarding that. Okay. Alright. So now we finally finish Kingdom Monera with two small headlines. Yeah, it is very small. One organism is Rickettsia and other is Chlamydia. We have to write 2 sentences only, and with that we will finish Kingdom Monera. Okay? With that we will finish Kingdom Monera. sterility in human males, pleuropneumonia of cattle. So now I am putting mycoplasma is over. Now rickettsia. Rickettsia. Please finish and then we'll move on. Everybody has finished it now? All right. Rickettsia actually what happened was there was a scientist named Ricketts. It was discovered by Ricketts while studying Rocky Mountains spotted fever. Ricketts while studying Rocky Mountain fever or yellow fever or Rocky Mountains spotted fever. Spotted fever. Rickettsia are a small group of gram-negative bacteria. These are different from other organisms, and they were discovered later on. So what happened was Rickettsia was discovered by Ricketts, and what happened was while he was studying this disease, he died because of Rickettsia. So this organism was named in his honor. It is transmitted by the bites of ticks, fleas, and very small organisms, ticks, fleas, mites, bugs, bed bugs, and all that. Okay, so it was studied by him, and because he was killed by that disease, so this organism was named after him. Okay, it's a potentially Rocky Mountain spotted fever is a fatal disease, lethal disease. Okay, so only one question is asked from it. Rickettsia in Rocky Mountain spotted fever. Rocky Mountain fever is transmitted by the bite of ticks. Ticks are very small organisms. So normally you can find them in people who live close to the hot tropical or hot dry climate areas. And if they don't actually wash their bed sheets properly, ticks will grow in number. Okay, so normally ticks are transmitted through cattle because they live in most unhygienic conditions, and they can also be passed on to humans. Bite of ticks, and it is potentially fatal disease. A person requires emergency medical care. Potentially fatal disease. Potentially fatal disease. Okay. Caused by Rickettsia rickettsii. Yeah, it's a lot of synonyms. Rickettsia rickettsii Okay, spread by the bite of ticks. Rickettsia also cause, um, it's also called Brazilian spotted fever, and it's normally not found in our country. It's very common in the Western Hemisphere. Western Hemisphere, I mean Western countries. It is very common in Europe and USA. So they suffer from this. Okay. They also cause typhus fever. Typhus fever also caused by these organisms. These organisms. That is all about rickettsia. Then there is chlamydia. It's also included in kingdom Monera. Chlamydia, we have to just write a single line. It's an energy parasite. Chlamydia is an organism which is an energy parasite. It feeds on ATP. It is an intracellular. It lives inside the cell and feeds on ATP. Intracellular energy parasite or ATP parasite. ATP parasite. With this, your kingdom Monera is finally over. Now kingdom Monera is over. We can now move on to kingdom Protista. Okay. So please tell me as soon as you have finished copying. Okay. So you have finished copying, I believe. Okay. So now we move on to kingdom Protista. Please put the heading kingdom Protista. Kingdom Protista. So kingdom Protista is a kingdom of unicellular eukaryotic organisms. Unicellular eukaryotic organisms. These are the first unicellular eukaryotic organisms. Unicellular eukaryotic organisms. Okay. They are the first unicellular eukaryotic organisms. And what happens regarding these organisms? Actually it's a cocktail of many different kinds of organisms. They are very diverse organisms. Actually this kingdom is so difficult to understand that it's been called a kingdom without proper boundaries. The boundaries of kingdom Protista are not well defined at all. The boundaries of kingdom Protista, they are not well defined at all. You can see here, the boundaries of kingdom Protista are not well defined. The boundaries of kingdom Protista are not well defined. Are not well defined. these kingdoms' boundaries are not well defined. All right. And why it is so? So let me explain why I'm stating this statement. Organisms that could have been placed in kingdom kingdom Plantae, algae-like organisms. Dinoflagellates, diatoms, they are placed in this kingdom. Organisms that are similar to fungi, the slime molds, they are placed in this kingdom. Organisms that are similar to animals, the protozoans, they are placed in this kingdom. So it's a different cocktail of diverse types of organisms. Are you getting me? Are not well defined because the diversity of this kingdom includes organisms which could have been a part of other kingdoms. So this is a very diverse kingdom. Therefore, we can say the food habit or the mode of nutrition, it is most diverse in kingdom Protista. The mode of nutrition is most diverse in kingdom Protista. So these organisms of this group are autotrophic, heterotrophic, as well as mixotrophic. So maximum nutritional diversity found in kingdom Protista, not in Monera, kingdom Protista. Okay? All right. Next point. These organisms are found in land, water, sea, etc. These organisms are found in land, found in fresh water, sea water, fresh water, land, air, etc. Also found in air, so quite commonly they can be cosmopolitan, but mostly they are found in land and air. Okay, so land and water, sorry, not in air. Air, they can be spores present in air, but they are not too common. Normally they require aquatic mode. All right. So kingdom Protista. Also, you can say this kingdom forms a link with fungi, plants and animals. Fungi, plants and animals. Not with bacteria, but with all the three kingdoms other than bacteria. Okay. Now, why are they eukaryotic? Because they have a well-defined nucleus. The nucleus is membrane bound and it is well-defined. So, please write. The organisms of this group, the organisms of kingdom Protista kingdom have a well-defined nucleus. And membrane bound cell organelles. well defined nucleus and membrane bound cell organelles. Membrane bound cell organelles. Okay, so they also can have cilia, flagella. Mode of reproduction is sexual, asexual. Okay, all right. Next point. Right. Okay. Next point. Locomotory organelles may be present. Locomotory organelles, that is cilia or flagella, may be present. May be present. Okay, and they can reproduce sexual as well as asexual, asexually as well as sexually. Protists reproduce asexually as well as sexually. By cell fusion and zygote formation. This is sexual reproduction and zygote formation. Zygote does not form in asexual reproduction. Okay. All right. Next, regarding chrysophyta. Chrysophyta. Okay. Chrysophyta are also called golden algae and diatoms. So please write Protista, like we are studying Protista, and Chrysophyta is a subdivision. Okay. So please write. This group includes golden algae and diatoms. Desmids or golden algae and diatoms. Okay. They are found in fresh water as well as marine environments. They are found in fresh water as well as marine water. Mainly in marine water. Fresh water specimens are quite less. Marine water specimens are more. Okay. And they are microscopic. Next point. They are very small organisms. They are microscopic and basically what happens is they can float in water, although they are very heavy. They are very heavy. They have a cell wall which is having silica particles, glass-like particles embedded in their structure. They are microscopic and float passively in water currents. When something is floating on the surface of water, it can be either animal material or plant material. So if it is animal material, it is called zooplankton. If it is plant material, it is called phytoplankton. Diatoms are the majority portion of all the plankton found in the world oceans. It is a main component of the plankton found all around the world. Float passively in water currents. This is called plankton. Okay. Now most diatoms are photosynthetic. They are photosynthetic. They are photosynthetic. And because they are part of planktons, they are major photosynthesizers in marine environment. Plankton, out of which diatom is a majority portion, it is the most important photosynthesizer around the world. They are the major photosynthesizers in marine environment. They are the major photosynthesizers in marine environment. Diatoms. They are the most important photosynthesizers among all the plant groups that exist in the world. Okay. Now, what do the diatoms look like? They are really, diatoms are very beautiful organisms, extremely beautiful organisms. So if you Google it, I'll send you a few images when I send you the PowerPoint presentation. Diatoms, they are really very beautiful organisms. When you look at them under the microscope, they look like diamonds, even more beautiful than diamonds, we can say, because they have the ability to refract light through silica particles embedded in their cell wall. Because of this reason, they are called jewels of the plant kingdom. Okay. So I'm going to ask you, have you seen diatoms around you? Yes or no? Maybe you have not seen living diatoms, but surely you are using some products that have diatoms in dead form. So can you guess the products of diatoms? They shine very brightly. They shine very brightly and they show a physical phenomenon called a total internal reflection. So have you seen any such thing? Which toothpaste? Not exactly, not in the toothpaste. There is something else. The girls might know it better. The girls might know it better. Boys also know it. Let me see if you can guess. Okay, I'll tell you. Lately, there are some makeup products there in the market. I know because my sister uses them. So there are there's something like some glitter particles like things, and they are put underneath the eyes, I believe, or over the eyes. I'm not sure. They are put somewhere on the face, I don't have the exact, yeah, it's puts over the face. I have seen that. So they have very shiny particles, very small particles, and this is one thing that it is used in. Other thing in lipsticks and in various makeup products, you can see very fine particles that are present in the texture of that product. Okay, these are made of diatoms. Diatoms have a cell wall which has silica. When the diatoms die, they sink to the ocean bottom, and then they are, after millions of years, it becomes a very hard rock. This hard rock is called kieselguhr or diatomite. Alfred Nobel made dynamite out of this. Previously, nitroglycerin was a very explosive substance and it could not be used by anybody. For example, if a truck was carrying nitroglycerin and it suddenly met a small bump in the road, immediately the truck would go to blast. So it was not able to be used until unless Alfred Nobel discovered that powdered diatoms called diatomite, they can be used to absorb their nitroglycerin, and it becomes a substance called the dynamite as we know it. So Alfred Nobel was instrumental in making a fortune out of it, and it was because of him that the Nobel Prize was instituted. Regarding the Alfred Nobel's discovery, diatomite is used in many modern discoveries today. For example, the night visible paint used in traffic light signals, the glow sign paint, it uses the diatoms kieselguhr. So it has a property that whenever light is falling on it in one direction, it can be scattered to multiple directions. So because of this property, diatomite or kieselguhr, that is used to make night visible paint, used in making cosmetics, used in making glitter paint, and in making dynamite also. Still today, dynamite is being made by kieselguhr only. Also in filtration of oils and syrups. Okay, so these are the various products. I hope you get the fair idea. Also, you people must be wearing some track suits, and when light falls on it in one direction, you can see that the light is reflected in multiple ways, right? So traffic people, traffic signal policemen wear some jackets or something. They also have these lines over them. On shoes like Reebok, Adidas, Nike, these strips are there. So this is made from diatoms. Okay, I hope you understand what I'm trying to say. Everybody has seen this night visible paints on the roads. Yeah, whenever light is falling on it from one direction only, it will be scattered to all the possible directions. So everybody can see that signal. That is the important property of diatoms kieselguhr. So please write now. Diatoms have an outer cell wall have an outer cell wall made of two halves each half fits the other like a soap box. Soap box. Like a soap box. So this is how diatoms are arranged. Two halves of a soap box structure. Right? This is how diatoms are arranged. Okay. So they have two unequal halves. Each shell is called frustule. Okay. And on this cell wall, there are many shiny particles called silica particles. Silica particles are attached. SiO2. silicon dioxide. That's a matter similar to in which quartz is made. Okay. The cell wall is encrusted with SiO2 particles. This causes diatoms to refract light brilliantly. Okay, so this causes them to refract the light brilliantly. Therefore, they are also called jewels of the plant kingdom. This question has been asked. What group of organisms is referred to as jewels of the plant kingdom? So actually it is not belonging to plant kingdom. It is placed in kingdom Protista. Still we call it jewels of plant kingdom. So people get confused that there is no alga like this and it is not in plant kingdom. So it's a confusing question, but the correct answer is diatoms. They are also called jewels of the plant kingdom. Jewels of the plant kingdom. So next time you see these cosmetics, take a look carefully. You're looking at the powdered remains of diatoms. After their death, diatoms sink to the ocean bottom. And over millions of years, they form huge deposits called diatomaceous earth or diatomite or Kieselguhr. Okay, and this diatomite is used in making dynamite, filtering of oils and syrups, Okay, and what else? Making cosmetics, night visible paint, etc. It's used extensively throughout the world. There are nearly limitless applications of this product. So it's using in making these things particularly. All right. So these are the products that you need to know about. Dynamite was made by Alfred Nobel. I'll just write also. Alfred Nobel absorbed nitroglycerin Nitroglycerin is a very explosive product. Nitroglycerin. It is made by reacting nitric acid with glycerin. Triple nitration of nitroglycerin gives nitroglycerin, a very highly explosive product. Nitroglycerin in diatomite to make a safe explosive. In diatomite to make a very safe explosive. Nitroglycerin is unsafe explosive. Safe explosive called dynamite. So dynamite was very highly used in making Suez Canal, Panama Canal, roads, bridges throughout the world, and railroads, etc. So whenever there was blasting of rocks that was required, it was done by the help of dynamite. So this was done by him, first of all. He discovered it, he made it, and he made a fortune out of it. Out of that fortune, the interest that accumulates every year, it is used in giving the Nobel Prize. Okay, so he made a fortune out of the Nobel Prize money is derived from dynamite, sale of dynamite to all the construction companies around the world during before 1900. Okay, so that is the thing. Moving on, now we have to complete about dinoflagellates. Please put the heading. Dinoflagellates. So dinoflagellates I won't be able to finish today, but let's begin it. What are dinoflagellates? Dinoflagellates are large organisms having two flagella. One flagella is transverse, other is longitudinal. Both flagella beat in their separate directions. So net result becomes that if we consider it as a vector, so there is a net movement because of the two flagella, but it is not in a straight direction. So it is in a curling direction, a top-like direction. So they are also called whirling whips. They move like in a helical fashion, rotating like this. Dinoflagellates do not have a straight movement. Also, dinoflagellates on the outside, they are covered with very thick plates of cellulose and pectin. They have two flagella, one flagella like this, other flagella like this. And on the outside they are covered with thick plates of cellulose and pectin. Right? This is a horizontal groove and vertical groove. Two flagella, and they are covered with thick cell plates. So they are also called armored protists. Longitudinal or vertical flagellum, this is the transverse flagellum. This is the transverse groove. Each flagella is kept inside its own separate groove. So let's write a few points and then I'll let you go. I understand you're getting late also. Okay, so they are covered with, they are also called armored protists. Armored protists because of presence of thick cell plates. Thick cell plates. Okay. All right, so it's about time. Finish this and then I'll let you go. Just this, finish this, and because it will take further time if I write further, you will get late. So I'm really concerned that I finish my class on time and you also reach home on time. I won't like to keep you further. So finish the students and then we can, then you can start leaving. Okay, tell me if you have done it. All right. Okay, so just don't be very much concerned. Just draw almost a rectangular figure. Make some arrangements on it. If you're not able to do it, I'll start from here also. I will draw the diagram again. Just write the thing that I have. Just write the points. Don't be concerned with the diagram. I'll have to draw the diagram again when I begin this class again. Okay, so I'll draw it. I'll start from here only. Just write the points. Okay, have you done it? All right. So I think I'll see you in the next class now and next week. So I'll be, in the meantime, I'll be giving you some PPTs related to this. I'll give you these notes also, and the PPT will contain the information regarding to these biological kingdoms and their description and a lot of images also. So it will help you in understanding what I have just taught you. So thank you, dear students. Good night, and travel back home safely. Okay, I'll see you in the next class. Thank you. Good night.